Hello there, my name is Vadim Pisarenco. I'm doing my PhD thesis at the Genetics Department of University of Barcelona in Evolutionary Genomics and Bioinformatics research group. Today, I'm going to explain you what my thesis is about. But before starting, I want just to say that I have the honor of being involved in the study of one of the most exciting organisms that have ever existed on our planet, and those are spiders. But before landing on the details, I'm going to give you 3 eye-opening ideas about why it is so important to study those organisms. First, they are ancient and old, essential in sustaining almost all known ecosystems by playing both roles of predators and prey. Second, they keep an eye on us because they hunt insects that may transmit diseases or cause plagues. Third, spider components such as venoms or silk have long been a source of inspiration for biotechnology and medicine because of their surprisingly versatile properties. So, you know the whys now. The next question is, how do we perform these studies? My thesis is focused on Dysdera spiders that have managed to colonize highly diverse environments, both on continents and islands, by adapting their morphology and diet. The big question in this thesis is, what happened during the transition from continent to Canary Islands? We know that the colonization and adaptation to new environments is quite often accompanied by changes in genome size, architecture, and even entire groups of related genes, also known as gene families. In this case, the genome size is much smaller on islands, and we suspect that the gene families from taste and olfactory systems must have played an important role in the process of adaptation, since they are absolutely essential for the interaction with the environment. Now, let's see what the process is to solve this puzzle. Well, first and foremost, what we need is a solid and stable foundation to start building on. That can only be achieved by exploring spiders' biology and genome. So, the first step is to sequence the DNA and update the genome. This can be understood as reading and reconstructing a strip book. This is a tough process, especially in spiders that are a really hard nut to crack. The second step is to find genes, those functional components, as well as other elements that form and shape the genome architecture. This is our foundation. Now the analysis can start. Genomes from several spider species are going to be compared and explored to understand what evolutionary processes and specific genome features were evolved in the transition to islands. This will add knowledge from a molecular evolution perspective and improve our understanding about those fascinating organisms. So just to finish, next time you see a spider, keep calm and think about their importance, complexity and beauty. Thank you.